In this week's lecture, we're going to be covering sensation and perception as a whole, but also focusing on one of the most important senses that the human beings utilize, which is the sense of vision. Next week, we'll be touching on other two senses that aren't as important, but just interesting to study as well. So we aren't, and you aren't, probably aware of any moment-by-moment work of our sensory systems. The nervous system actually is getting tons of sensory input at the moment from your eyes, from your skin, from your ears, and you're just not aware of all the work that your brain is doing to filter out that information, allowing you instead to just focus on my voice as well as the video that you're watching. So the brain's getting a ton of information at the same time, and incredibly it integrates all the sensory information into one coherent story. So for example, you're probably not paying attention to the clothes on your body right now as you're listening to me talk about this material and watch this video. But now that I've called your attention to it, you're probably more mindful about the clothes touching the skin on your body. So most of the time, the sensory information that we're getting into our brain is not something we pay attention to, but the brain is nonetheless getting that information. What's incredible is that our brain, remember, our brain can only understand in action potentials and changes in membrane potential, but our brain is capable of making sense of different types of stimuli, right? Sound waves from our ears, light from our eyes, pressure from our skin, as well as odorant molecules. What we'll be talking about for this class, for this week and next week, is that their sense organs, which are actually neurons, converts all of these different stimuli into a neural impulse that then gets sent to the brain. So to me, that's pretty darn incredible that you can take information in all of these different forms and convert it into a language that the brain understands. So humans are especially equipped for the sense of vision. So 30% of our cortex is devoted to processing the sense of vision, which is why the story of Daniel Kish, shown here on a bike, is so incredibly remarkable. So he lost sight in both eyes when he developed an aggressive form of cancer that affects the back of the eye we'll be talking about in this lecture, called the retina, to protect him. So to prevent the cancer from spreading, they removed his real eyes at the age of 13 months, so he was barely a year old when he lost his sense of vision. But he is able to navigate a street on a bike easily, as he is doing here. So in this picture, he has no sense of vision. The way he does it is by producing clicking sounds with his mouth so that his ears can pick up any sound that bounces off of objects that are in front of him, to the side of him, and he's able to create a visual picture of his world using those clicks without his sense of vision. His brain has adapted to no vision by heightening the other senses, right? They've reallocated the space that would have been set aside for vision for other senses, allowing Daniel, incredibly, to navigate his world using other sensory systems like hearing. So as I mentioned earlier, 30% of our cortex is devoted to decoding light into images and complex scenes, but some animals have incredible visual capabilities compared to human beings, right? So we think we have good vision, but think about an animal like the hawk, for example. Many species surpass us in the strength of vision, but also in the strength of other traditional senses as well, like smell, touch, and sound. They're much better at detecting those sensory stimuli than we are as human beings. But because we rely so heavily on our sense of vision to gather information from our world, we as a class are going to devote the entire week to this particular sense, and then we're going to transition to other senses next week. So I don't know if you guys got the Marvel reference here with respect to vision. I'll let you figure that out. Alright, so all sensory systems share common characteristics. an operating principle. So sensation and perception, if you took enteropsych and heard about it at any point, actually form the basis of psychology, right? So biologists were curious about how human beings experienced the world to get a better idea of what was going on in the mind. And so sensation and perception became the first research studies behind psychology. But sensation and perception are critical for us as a species. So for effective and adaptive behavior, the nervous system needs to be really good at detecting information from the environment, which is what sensation is, but it also needs to be able to interpret that information or process that information, which is what perception is. So sensation, detection of the information, is going to happen in the sense organs, but perception is actually happening at the level of the brain. So in 1850, this gentleman here, Gustav Fechner, had a flash of insight as he laid in bed one morning. And so most individuals have these brilliant ideas when they least expect it. And he had it right before he was going to bed, or he was waking up in the morning. So the insight that he had caused him to consider this intersection between the mind and the physical world by measuring a person's perception in response to changing physical stimuli, material stimuli. And so was born the field of psychophysics and the beginning of psychology. So when an experimenter gradually and systematically increases the intensity of light from an extremely dim luminescence, shown here, to one that the subject will eventually report a change in intensity, this is known as the just noticeable difference. So the point at which the subject is able to detect a difference between one stimulus and another is the just noticeable difference. So it's the minimum amount a stimulus must change for a subject to detect a difference, regardless of whether the stimulus is light, sound, or pressure or heat. So in this example that I'm showing you, it's light, but in other cases, early psychologists were testing other stimuli like sound as well. So that's called the just noticeable difference, or the difference threshold. So the difference threshold and just noticeable difference are the same. Research showed back then, even, and still holding true today, showed that the just noticeable difference was a constant proportion of the initial stimulus. It could be mathematically calculated. So in contrast, the absolute threshold is the lowest intensity of a stimulus a person can detect 50% of the time, and it might be that it's this particular luminescence here. But again, it's going to vary from person to person, but the first visible stimulus of light or sound, and you might have actually heard, if you had the hearing test done in school or at the doctor's office when you were younger, they produced a sound that you could barely hear. That is called the absolute threshold. The first time you can hear any sort of sound. And as you get older, the absolute threshold changes because your hearing gets worse, your eyesight gets worse, unfortunately. So the absolute threshold shown here is the lowest intensity of a stimulus a person can detect 50% of the time, which is what I just said. So as animals began to live in various habitats over the course of evolution, a vast array of sensory adaptations evolved. So we have the star-nosed mole rat here that is completely blind, but uses this incredible organ that's actually outfitted to its face that's very sensitive to touch. So it tends to burrow, and the way it senses its environment is by touch through this incredible organ here. Dogs have an incredible sense of smell as well as hearing. Hawks, we talked about earlier, have amazing vision. Honeybees, for example, can see light at wavelengths that we are not able to see. So they can see UV light, and we would need like special goggles to be able to see that. So the real world sensory experiences are not experienced by individual sensory systems, but rather are influenced by the simultaneous integration of many sensory systems. So when we experience the world, we experience it not solely by vision, but by what we're feeling on our skin, by what we're smelling. And so multi-sensory systems can converge, for example, on the sense of smell. Esther Stenbergs went to a cool place in Philadelphia that I wish I'd known about because I lived near Philly for, a good chunk of my life. But there's a place called the Monell Chemical Senses Center where they actually spend time. They have research scientists studying chemical senses. And so she participated in an experiment in which she placed her face in a chamber containing a single aroma as she looked at different scenes. So in the first scene, she smelled that aroma and saw a beautiful mountainous scene. She described, when she was asked to describe the aroma, she said that the aroma smelled very pleasant and fresh. In the next portion of this experiment, she did the same thing. So she stuck her face in the chamber and she smelled that single aroma, but this time she saw a picture with a city with smokestacks that were expelling horrible, gross, dark smoke. And she described that aroma as being toxic and pungent and gross. I'm sure you probably guessed that the aromas in both those conditions were identical. Her interpretation, Esther's interpretation and perception of the aroma, was influenced by the scene that she was viewing at the time. So her olfactory perception was influenced by cross-modal stimuli from two senses. In this case, it was her sense of vision. So another example of a multisensory processing is the way smell, taste, and flavor converge. So what you're looking at here are the sense organs for taste. So taste is actually detected by taste cells that are found on our tongue. But it's also influenced by our sense of smell. So the perception of flavor that we often think of as originating from our tongue and our mouth is based on the convergence of cues from several senses. We also use our sense of vision to look at the slice of pizza that we're biting into, the smell of the pizza, the taste, as well as the texture of the pizza in our mouth. So taste buds, as I mentioned, are these small, where's my clicker? My pointer. Taste buds, as I was mentioning earlier, are these small structures of soft tissue located deep embedded in the tongue and in the soft palate as well as areas of the throat. And they contain receptors, specialized cells, for detecting the different types of tastes: sweet, salty, sour, bitter, as well as the savory umami flavor. And scientists have determined that several areas of the primate cortex are involved in multisensory integration. So in this particular diagram, you're seeing various areas of the brain that integrate multiple sensory input. So take, for example, the superior temporal sulcus, which can be found in the temporal lobe of the primate brain. So this particular brain area is involved in vision and hearing correspondence, so essentially speech. So it's like this area is involved in integrating synchronization between lip movements and speech sounds. The McGurk effect demonstrates when these two senses, sight and sound, are not synchronized. So if you were hearing the sounds for bows, but seeing the lip movements for the word goes, you would actually perceive something that's consistent with the visual input, and you would think you heard the word doze or those, okay? Even though the sound was bows. So this perceptual experience occurs because the brain's cross-modal system, like the superior temporal sulcus, is attempting to correct this discrepancy between what you're seeing and what you're hearing. So you have to think about senses as this integrative system, and we're not going to really focus on the integrative system, but you might be interested in learning more about it, so I wanted to share it with you. But for now, we're going to focus on one of the 5 sensory systems, the one that's most important to human beings, and that's the sense of vision. So as visual information travels from the eye to the brain, and then to higher visual processing centers, it gathers more relevant information to produce what will ultimately be a meaningful image for us. So light, which is what our eyes can see, is part of a larger electromagnetic spectrum shown here. But we can only see a very small portion of it. So the portion that we can actually see that's expanded down here is right here. So it's a very small fraction of that larger electromagnetic spectrum. The light that we can see can be binned or grouped into 3 categories. Shorter wavelengths, which typically are in the blues, medium wavelengths, which are the greens and yellows, and then the longer wavelengths, which tend to be, and you can't even see my marker, are red. But if you recall, our brain can only understand information in the form of action potentials or neurotransmitters. So incredibly, there are specialized cells in our eye that are responsible for converting light into an action potential before it gets to the brain. And that's what we're going to be spending the rest of this lecture talking about, the ways in which light gets converted into a neural impulse. So converting a sense into a neural impulse is called sensory transduction. But because we're focusing on the visual system, the conversion of a sense, in this case light, into a neural signal is called phototransduction, or your textbook calls it visual transduction. Either way, it's referring to, the conversion of light into a neural impulse. So what in the eye is responsible for seeing or for this process of phototransduction? So when light enters the eye through the pupil, which is this hole that you see here, and it's usually the black portion of the person's eye in the center of their eye, the size of the pupil is determined by the muscles around this pupil called the iris. It's what gives us our eye color. Light is focused on the back of the eye as it travels through the lens. So this structure here is the lens, and its size determines where the image gets focused depending on how far away the image is from us. And we'll talk about the fovea in a second, but it is focused to the back of the eye, which is the seeing portion of the eye called the retina, but more specifically it's focused onto the fovea, which we'll talk more about later on. So let's talk about what the retina is made up of. So what you're seeing here is actually the retina blown up. It's made up of several distinct layers and types of cells. So the caudal-most layer is made up of photoreceptors, the ones that are actually responsible for detecting light. So they're the ones that are furthest away from the source of the light. The next in line are the bipolar cells shown here. They're the ones that get the information from the rods and cones. And then the amacrine cells and horizontal cells, together with the bipolar cells, send the information to the ganglion cells. So the ganglion cells are the ones that end up transmitting this information to the brain. So the ganglion cell axons are what make up the optic nerve that exits the eye to the brain. So because the optic nerve leaves the eye where it does, there's a portion of the retina that has a blind spot. It lacks photoreceptors, so there's no way for it to detect light. So if a portion of the eye doesn't have photoreceptors, we technically don't see it. There's several tests for it online, but even one in your textbook that I'll show you next, so you can check out to detect your blind spot. So what you're going to do is you're going to focus using only one eye on either this red pepper or yellow pepper. If you're going to focus on the red pepper, you want to use your right eye and close your left. if you're going to be focusing on the yellow pepper, you want to close your right eye and focus your left, and you're going to slowly bring your face to the screen, and what you'll notice is that the other pepper eventually disappears because that pepper ends up falling onto the blind spot of your retina, which doesn't contain any photoreceptors. So let's talk about these photoreceptor cells that are responsible for detecting light. So photoreceptors, rods and cones, differ in several distinct ways. So rods are prevalent in what's called the peripheral retina, so everything but the fovea. They're sensitive to low levels of light, so when we're in a dark room, we're largely using our rods to be able to see anything if we can see anything at all. They're monochromatic, that means they don't see color, and I don't know if you've noticed, but when you're in a very, very dimly lit room, it's really difficult to discern the shade of color that you might be able to see. Another reason why you might not be able to see very clearly is because rods have poor acuity, so they have very little attention to the ability to detect detail. Cones are most prevalent in the fovea. So fovea is that portion of your eye where the lens focuses the image onto when you're really looking at something. So right now, as you're looking at the screen of your phone or your computer, the lens is focusing the image, the words on the screen, onto the fovea so you can read it very clearly. It's sensitive to high levels of light and can gather color information. These cones that are jam-packed densely in your fovea are able to detect color, and they have excellent acuity, so they have sharp vision. They're able to discern lots of detail. So you're probably wondering why rods and cones are called what they are. Again, it'll be the theme of this course, but behavioral neuroscientists and neuroscientists in general are not very creative. But rods were given the name rods because of the shape of their cell body, and cones were given that name because of their shape as well. So they're called rods and cones because of what they look like. Okay, so as I mentioned, in addition to the rods and cones in the retina, there are other layers of cells that exist, and their functions differ. So bipolar cells connect photoreceptors to ganglion cells, because remember, the ganglion cells are the ones that eventually carry light information from the retina to the brain. The horizontal cells are found between the layers of photoreceptor cells and bipolar cells, and amacrine cells are found sandwiched between bipolar and ganglion cells. The point of the horizontal and amacrine cells are to transmit information across rather than through. And so when we talk about a process that's called lateral inhibition later in this week's lecture, so not this lecture but the second one, lateral inhibition, horizontal and amacrine cells are responsible for creating that visual phenomenon. So as light information travels from the retina to the brain, it travels in this order through the retinal cells. So it goes from the photoreceptor cells, which are responsible for converting light into a neural impulse. The bipolar cells respond to this information and then transmit it to the ganglion cells, and then it's the axons of the ganglion cells that actually send this information to the brain. So here's another kind of visual on how it happens. This is the back of the eye, and this is closer to the lens, and so light hits the photoreceptor cells, it gets transmitted to the bipolar cells, to the ganglion cells, and then to the brain, and then we've got the amacrine and horizontal cells kind of transmitting information between bipolar and between ganglion cells. So let's talk about how photoreceptor cells convert light into a neural impulse. So we're going to focus on rods rather than cones because rods are just able to see black and white. They either see light or they don't. So rods have two distinct parts. They have what's called an inner segment, shown here, and an outer segment, shown here. The inner segment is what contains all of the cell's machinery, the nucleus, the organelles, everything that it needs to keep the cell alive. But the part of the outer segment is the one that detects light. So the outer segment has these pancake-like discs sandwiched all throughout the segment called lamellae. And within these lamellae are pigments that respond to light. So they're called photopigments. Photopigments in rods are actually made up of two molecules, opsin and retinol, and collectively these two molecules are called rhodopsin. So the photopigment in cones is different than the photopigments in rods, but we're going to be focusing on the rods for the duration of this lecture. So rhodopsin is the photopigment, and it's made up of opsin and retinol. So the way in which phototransduction occurs in the rods is that when there's no light, so in the dark, this is the state of the lamellae in the rod. So this is rhodopsin right here, retinol and opsin, and in the dark there is a sodium channel, an ion channel that's open, and sodium is rushing into the cell, making the inside of the rod depolarized. So it, in the dark, is actively releasing neurotransmitters to shut down bipolar cells. So I'm going to erase this mark here so you can see how it changes once the light hits. So once the light hits the rhodopsin molecule, it bleaches, causing the retinol molecule to change shape, shown here by the change in color. And rhodopsin has a specialized protein attached to it, similar to what you know about metabotropic receptors. So it has a G protein attached to it called transducin. So when light changes the shape of retinol, it causes the opsin molecule to change shape as well, and for the G protein to come off the rhodopsin molecule. When the G protein comes off, it activates an enzyme called phosphodiesterase, abbreviated PDE here. What the PDE does is it breaks down this molecule called cyclic GMP. It breaks it down. And now the cyclic GMP molecule is needed to keep the sodium channel open. So when PDE breaks down the cyclic GMP, it causes the sodium channel to close, right? It causes the sodium channel to close. And when sodium no longer enters the cell, it causes the rod to become hyperpolarized and shut down. Okay? So if you didn't quite get these steps, I would go back and watch this portion of the lecture again and write them down. But in your textbook, it's giving you the different steps that occur in this particular figure. So again, light hits the rhodopsin molecule, activates transducin. Transducin's job is to activate PDE, phosphodiesterase. Phosphodiesterase breaks down cyclic GMP, which is what's keeping the sodium channels open. Once cyclic GMP is broken down, the sodium channel closes, causing the rod to become hyperpolarized. So that, in a nutshell, is what the figure from your textbook shows. But feel free to go back and watch this portion of the lecture again, because it's pretty complicated. It's got many steps in it, and I want to make sure that you understand the reasoning behind this, because this essentially is how light, right, which is an electromagnetic wave, gets converted into a neural impulse that the brain is able to understand. Light hyperpolarizes the rod. So remember, in the dark, the rod is releasing neurotransmitters onto the bipolar cells, keeping the bipolar cells shut down. So it's inhibiting the bipolar cells. When light hits the rods, it causes the rod to become hyperpolarized. It stops releasing neurotransmitters, and because the neurotransmitter used to shut down the bipolar cell, the absence of the neurotransmitter activates or depolarizes the bipolar cell. This causes the bipolar cell to release neurotransmitters onto the ganglion cells, and then the ganglion cells have action potentials which get sent to the brain. Okay? So it is this process that allows us to detect light. So I just want to put this into context again, your ability to watch this video to detect the light and the text on the screen is the result of the actions of these rods and cones that are in your eye right now, behaving the way they are and sending this information to the brain. So this is by far going to be the most complicated portion of this week's lectures. So make sure to go back and watch these portions again if you need it, if anything was confusing. But we're going to summarize this lecture for now. So sensation is the detection of a stimulus, and perception involves processing that stimulus, which allows for proper responses from our nervous system to environmental stimuli. Animals like humans have evolved sensory specializations that allow them to detect, touch and smell much better than human beings. Our brain integrates multiple sensory inputs to give, or to create rather, that's a typo, the perception of our world. Vision is the most important sense to human beings, and photo or visual transduction happens in the retina in specialized photoreceptors called rods and cones. A blind spot is created when the optic nerve, which are the axons of the retinal ganglion cells in our retina, exit the eye. Phototransduction in the rods causes them to shut off or hyperpolarize, which activates bipolar cells and ultimately activates retinal ganglion cells. So don't forget to submit the reflection for this particular lecture and watch part 2 as well.